This method can help answer key questions in the field of drug discovery to identify anti-cancer effects and gain better mechanistic insight into novel molecules using in vitro and in vivo 3D culture systems. The main advantage of this technique is that it validates the pharmacological effect of compounds in a more physiologically relevant in vivo model.
To begin, plate 20,000 A549 cells per square centimeter in 15 milliliters of rPMI 1640 cell culture medium, supplemented with 10% FBS and 1% antibiotics, in a 75 square centimeter flask. Incubate the culture at 37 degrees Celsius and 5% CO2. On the day of the experiment, use a hemocytometer to determine the cell concentration. Collect 50,000 cells in a 1.5 milliliter microcentrifuge tube by centrifugation at 400 G for 7 minutes, and resuspend the cells in 100 microliters of sterile 1X PBS. Using a multi-pipette, dispense 1.1 milliliters of semi-solid methylcellulose-based medium, or MCBM, into tubes, preparing 1 tube for each condition. Then, add 110 microliters of FBS to each tube. Next, seed 1,000 cells per milliliter. Then, pipette 2.4 microliters of the stock cell solution into the tubes of MCBM and FBS. After adding the cells, hold the tubes vertically and vortex them at the highest speed for 1 minute to thoroughly mix the MCBM and cells.